Let's go ahead and take a look at chapter 30, which is an introduction to animals. Not yet animal systems. We'll go ahead and get to that later on. Today will be, or this presentation will be about mostly defining what an animal is, the time period in which they originated, which was back 550 million years ago during the what's known as the Cambrian period. There was this phenomenon called the Cambrian explosion, which diversified widely animal species. We'll take a look at some of the innovations during the Cambrian explosion, and then we'll take a deeper look into the diversification within the animal phyla. We'll go ahead and differentiate between non-bilaterian animals and bilaterian animals, which are animals that show bilateral symmetry, and then we'll get into an introduction to protostomes, deuterostomes, their embryological development, and then we'll elaborate from there in other presentations. Let's start by taking a look at the phylogenetic tree where animals are located. Keep in mind, animals are eukaryotes, and they belong to, or they are located rather, within a larger, broader group or lineage called the Opisthokonta, together with fungi and choanoflagellates. So if, as you can see in the phylogeny there, animals share a common ancestor with the choanoflagellates, which occurred somewhere around 900 million years ago. So if the line represented time, then from here to here would be about 900 million years. Recall that eukaryotes are about 2.7 billion years old. Earth is about 3.7 billion years old. So just to kind of put things into perspective. So now, what is an animal? Yeah, so what defines an animal? First of all animals fall into this category, into this phylogeny there, which is a monophyletic group, and monophyletic groups contain their common ancestor and their common ancestor, which occurred right around there. And then all of the species within the animals which fall into that phylogeny there. So that would be a monophyletic group, and animals fall under that category. Now, what is an animal? What are the characteristics of an animal? Well, first and foremost, animals are multicellular eukaryotes. They have no cell walls, and they have an extensive extracellular matrix. So what does it mean to be a eukaryote? If you recall, a basic definition of being a eukaryote, having a true nucleus, which is a nucleus that holds DNA within a membrane-bound nucleus. Yeah, so there's a membrane that surrounds that nucleus and protects the DNA within chromosomes. So another characteristic of eukaryotes is that eukaryotes contain multiple membrane-bound organelles, whereas prokaryotes don't have either. Yeah, so they don't have a true nucleus. They do have DNA, but it's not surrounded by a membrane, and they do not have internal membranes, multiple internal membranes. They only have one plasma membrane on the outside. So again, no cell walls, but they do have an extensive extracellular matrix, so meaning that within the body you will find an extensive network right in between cells. So animals are made up, or a human is made up of at least 30 trillion cells. In between those cells, there are other things. There's other tissue, like for example, connective tissue that connects those cells in some way. There's a lot more tissues and we'll deal with those in other presentations, but for now, the extracellular matrix is that space in between all of the cells. Other characteristics that make up animals are they use proteins for cell-to-cell adhesion. So for example, glycoproteins that hold cells together, and the specific types of tissues. They are heterotrophs, or we are heterotrophs, meaning that we must kill to consume and form our energy in that manner. They move under their own power at some point in their life. Keep in mind that most of our life we move under our all of our life we move under our own power after birth, that is. But some animals don't. So if at some point in their life they move under their own power, then they fall under animals, the animal category. The animals do have a nervous system which works together with the muscular system in order to achieve movement. So here are the major animal phyla, which is there's about 30 to 35 phyla of animals. They're all divided up into a few smaller groups. First and foremost, the non-bilaterian groups, which includes all of these phyla and these many species. Alright, so non-bilaterians are characterized by the fact that they do not manifest bilateral symmetry in their bodies. And then we have the protostomes, which are divided up into two smaller groups. Protostomes, again, would be divided up into the Lophotrochozoa, which is right here. These are all the phyla within the Lophotrochozoa group, and look at how many species there are within it. The Ecdysozoa would be another smaller group for protostomes, and these many species exist within the Ecdysozoa. Now, protostomes are called protostomes. Their name comes from their embryonic development. I'll go over this in more detail later in the lecture. But in summary, during embryological development, their mouth develops first from what's called a blastophore. A blastophore is basically an indentation. It's a folding that originates from what's called a gastrula. A gastrula is essentially a later developmental stage from a zygote. Keep in mind, zygotes are made from the fertilization of sperm to an egg. So after fertilization, there's many different stages of development there. One of those, again, is called the gastrula stage. The gastrula creates a blastopore, and this blastopore can either become a mouth in protostomes, or it can become an anus in deuterostome animals. Again, I'll go over more details in a little bit. Now, deuterostome animals, there's three phyla within it: the Hemichordata, Echinodermata, and the Chordata. So we fall under the Chordata phyla. As you can see there on the bottom, we are mammals, and therefore fall under the Chordata phylum. So what makes the Chordata a Chordata? Well, it's a few characteristics, but first and foremost, it would be the existence of a dorsal nerve cord, essentially. So a cord that gives rise to many nerves. So here we're looking at all of the animals in another version of the same phylogeny I showed you earlier. So again, the entire Animalia. Here we have the choanoflagellates, which are the most related species, closely related to animals. They don't look like animals, but nevertheless they are closely related, at least genetically. So Here are all the animals and their groups. This is the bilateria, the non-bilaterians, protostomes with their two major groups, Lophotrichozoa, the ecdysozoa, and then the deuterostomes here on the bottom, and the chordata, that's where we fall under, and all mammals for that matter. But keep in mind, from left to right, the time period is about 900 million years that spanned from multicellularity all the way to essentially now. So here is the point where multicellularity originated. Here's a common ancestor which gave rise to Porifera and everything else within the animals. By diploblasty, or the development of diploblasty, allowed some of these animals to develop embryologically from two germ layers, the ectoderm and the endoderm. I'll go over more details later. What's important to us is that we are triploblast, meaning that we develop from essentially an embryo that contains three germ layers: the ectoderm, the mesoderm, and the endoderm. Ecto outside, a layer outside of the organism. Meso means in the middle, and then endo means inside. Yeah, so three germ layers. So how have biologists been able to infer all of this data? So, we were not in existence 900 million years ago, evidently. We've only been here anywhere from 200,000 to 300,000 years. So how do we know all these things, right? Well, from evidence. We've already studied this evidence when we were talking about evolution. But just a little bit of a recap here. Fossils, first and foremost. These are leftovers or remains of the hard parts of animals. Excuse me. Mostly bones. There's also comparative morphology or synapomorphies, but the body plan, the shared similarities between anatomical similarities between species. There's also comparative development, which refers to embryological development and the similarities between different species. And also comparative genomics, which nowadays is they're all big fields, but nowadays genomics is increasingly and progressively more important because of the advent of technologies. We're able to analyze DNA using all sorts of techniques and strategies. And again, comparative genomics, or the comparison between DNA and RNA from different species and proteins for that matter. This is how essentially we're able to figure out the similarities and phylogenies between different species of the animal phylogeny. So let's go into a bit more detail in terms of the different innovations that animals showed ever since their origin during the Cambrian explosion 550 million years ago. First and foremost, their development. Yeah, so we're looking at a diploblast animal right now, and this is evidently early in embryonic development. Here's what I meant by endoderm. The endoderm would be the inside layer of cells, or the inside tissue layer. Then the ectoderm would be located outside. This is a diploblast animal. Now, there's also triploblastic animals, meaning that they have three layers. So you're looking at two layers here, and then there would be another layer right in between the two layers, and that's called a mesoderm. As I mentioned, these layers develop after fertilization. Yeah, so a few hours to a few days after fertilization, actually a few weeks, I'm sorry. After fertilization, we have the development of these three germ layers. Keep in mind that in order for animals to reproduce, right, and create the creation of a new animal, entails a sperm and an egg. So those two, at least a triploblast animal, those two coming together create a zygote, and that zygote is essentially a one large cell. So you have an egg cell, which is really large, and then you have a sperm cell, which is a lot smaller than an egg cell. Those two come together, they combine their chromosomes, their DNA, their genomes, and they create a zygote. The zygote is a large cell with, at least in animals, at least in humans, with 46 chromosomes. This large cell starts dividing into 2 cells first, and then 4, 8, 16, 32, 64, and so on, until we have enough cells to be able to make this structure here. Again, it happens within a few weeks of embryonic development. So this diploblast and triploblast, for that matter, contains the entire genetic toolkit in order to make all of the organs that the organism will have for the duration of their life. So just a bit of a summary here. The ectoderm, which is the outside layer, gives rise to the skin and the nervous system of an organism. The endoderm, which is the inside layer, gives rise to the lining of the gastrointestinal tract, which is everything related to digestion. And then the mesoderm is going to give rise to everything in between. So you have the GI tract in the middle, you have the skin on the outside, and then everything in between, which is the organs, the muscles, bones, internal structures, that is again given rise to by the mesoderm. And then you have evidently from the mesoderm you have the development of muscles. Muscles are made up of contractile proteins known as actin and myosin. These are contractile proteins that slide essentially across each other, on top of each other, and they manage to contract muscles, and that it just turns out that's a good adaptation for movement. So later on in development, actually when mature animals manifest different kinds of body symmetry, different kinds of body plans. Let's go ahead and start off with radial symmetry, which is symmetry on essentially on two planes. So you have your cnidarians showing radial symmetry, and again this is symmetry on 2 planes. Just 1, 2, and so on. So bilateral symmetry, which is what we are mostly interested in here in this class, is basically symmetry on one plane, such as is shown here in this diagram. Yeah, so these two parts are basically mirror images of each other, and these animals are going to be known as bilateral animals. Everything else would be a non-bilaterian, whereas these are bilaterian or bilateral animals. So now all triploblasts, we are triploblasts, all mammals are triploblasts. They are all bilaterians, and we do contain within our genetic toolkit a group of genes called HOX genes, HOX genes. You might want to read up on those, which are essentially genes that regulate our body plan. Yeah, so these again allow us to develop from anterior to posterior, or front to back, and then dorsal to ventral. Yeah, so again, body plan genes, or HOX genes, regulate the body symmetry of essentially any animal. Now, extending on body symmetry, it's important to talk about body symmetry because of the organs that animals contain, all of their anatomical structures. To include their nervous system. Body symmetry and nervous system are associated. They go essentially hand in hand. So there's different manifestations of nervous systems throughout the animals. We're looking at two. We're looking at nerve nets found in neurons in hydra. As you can tell here, there's no brain, there's no central nervous system. There is essentially just a nerve net, which shows a high degree of interconnectedness. Keep in mind, nervous systems transmit and process electrical signals, yes, and they're all made up of neurons, including nerve nets and our central nervous system. So again, this is another variation of nervous systems in animals. It contains a central nervous system. And then within us, in humans and mammals, there's a lot more parts to the nervous system, a lot more divisions and a lot more functions. So larger organisms require a highly diverse and highly evolved nervous system. So other variations of nervous system aside from nerve nets and CNS are clusters of nerves. There's also ganglia. There's neuronal tracts, cords, ganglia. We happen to have all of those but nerve nets. Central nervous systems contain several parts. So in mammals, they are a brain and a spinal cord, and then from the spinal cord we develop nerves, and you can see them here in this worm. They're not as developed as ours, but nevertheless they do contain some sort of brain, ganglia, and nerve tracts, which give rise to nerves. Another innovation within animals is the coelom. Yeah, so coelom or coelomates are characterized by having a coelom, and what a coelom is, and you're looking at it here in this worm diagram, is essentially the space which is fluid filled right in between the gut or the GI tract and the outside of the animal. So this space it serves different functions. For example, in this worm, the coelom is full of water, and mostly water and nutrients, and its function is to act as a hydrostatic skeleton, which is going to give support to the animal, and it's going to go ahead and allow it to move. Our coelom in mammals has different functions. So it holds all of the organs, all of the extracellular matrix. It's still the space in between the GI tract and the outside of the animal or the mammal. It holds all of the organs. It is fluid filled. It allows for oxygen and nutrient circulation in us, and it gives our organs a certain degree of flexibility of movement. Recall, and we don't think of this because of our autonomic nervous system, but all of our organs are in motion. Yeah, so all of our organs move. Our lungs move, our heart moves, the digestive tract moves. So again, the coelom allows for a certain degree of flexibility and movement to organs. So let me elaborate a bit on the formation of the germ layers in animals. We're looking at 3 germ layers here. So this animal is a triploblast animal, but keep in mind there's also diploblast animals with 2 germ layers. So what I'm showing here is the different stages of development or embryological development. There's not even an embryo yet. Right at this point, we have the formation of a gastrula, which is essentially a ball of cells which have not differentiated yet, or even formed the 3 layers. It's barely starting to form the 3 germ layers. So there's no organs yet. There's nothing yet. This would be about week 2 to week 3, give or take, in development. This is not the beginning of development. Keep in mind the beginning is called a zygote, and the zygote is one cell thick. But regardless, that one cell would maintain its size. So this is about the size, more or less, of the zygote. And it still continues to maintain the same size for about 2 to 3 weeks until the 3 germ layers are fully formed. And then from there, then you have the formation of the actual animal, segmentation of some animals, which gives rise to specialized adaptations. But then other animals like mammals form their organs in a process called organogenesis. Yeah, so first and foremost, we have the vegetal hemisphere here right before we form the blastopore and the gastrula. The blastocele is a fluid-filled cavity inside of this ball of cells. It continues to divide. It forms a blastopore, which is essential to development of different animals. This blastopore is maintained throughout the days in development of the development of the animal, until the gastrula is formed, where the 3 germ layers, ectoderm, endoderm, and mesoderm, start to be formed. Notice what the blastocele is doing. This fluid-filled cavity is now indented. Yeah, so it's now indented. So this part right here underwent an indentation and a folding, right, to form that blastopore. And again, this blastopore is gonna go in different directions when it regards protostomes and deuterostomes. In protostomes, the blastopore is gonna go ahead and form the mouth first, and then from there, it's gonna go ahead and develop everything all the way down to the anus. So from mouth to anus in protostomes. In deuterostomes, it's going to be the other way around. This blastopore is going to go ahead and form the anus, and then from there all the way to the mouth. So that's essentially what differentiates deuterostomes from protostome animals. And evidently from there is you have their development. So after this, you would essentially get an embryo, which starts either segmenting or goes through organogenesis. But keep in mind, this is all regulated by this majorly important group of genes called the HOX genes. So throughout the animal lineage, you can see wide variation in the anatomical structures of all animals. There are body plans which are again modified and regulated by HOX genes. So what we're looking at here in this diagram is the development of different sensory organs, which gives rise to the senses in animals. These senses, as well as every other adaptation of animals, originated due to natural selection. Yeah, so keep in mind, natural selection acts on genes, yeah, within a gene pool within populations. So it's essentially changes in allele frequencies in populations which are advantageous to the reproduction, not only the survival of the species, but their reproduction. So these are again the different sensory organs. You know what they are, right? You have them in your body: sight, taste, smell, hearing, touch. We'll go over the details starting later on this week and also throughout next week, and we'll spend a good chunk of time talking about these sensory organs and all of the different body systems within mammals. So although the senses apply to all animals, the different parts of the animal lineage show even further variations in senses. For example, the ability to sense temperature in order to not only navigate the environment but also identify prey. There's also the ability to sense magnetic fields, for example, in sea turtles and actually birds. There's some new research within quantum biology that points to the direction that birds need to be able to sense the Earth's magnetic field in order to navigate. There's also the ability to sense electric fields, like in sharks, for example, to be able to sense prey. Birds also manifest the ability to sense barometric pressure or atmospheric pressure. And it's said to be important in order to avoid things like storms, environmental conditions that are not favorable for bird migration. And then yet other species are able to even sense gravity, right? So moving on to other innovations, animals also show variation in the ways and means to eat. So a means to obtain prey and food in order to make energy. Yeah, so there's 4 different types here. We have detrivores, herbivores, carnivores, omnivores. You've heard of these before. Just a summary real quick. Detrivores feed on organic matter found on within the soil. Keep in mind, organic matter is mostly made up of carbon, but evidently these animals also need other chemicals to be able to survive, like sodium, potassium, and so on. Herbivores feed mostly on plant tissue. They don't necessarily have to kill the plant. They can feed on parts of the plant without having to kill it. The plant can regrow those, either regrow their tissue or grow larger and branch out even more. Carnivores need to kill in order to eat, and then omnivores can feed off of a combination of both meat and plants. Again, things that you've already seen in the past. But then we go into the different forms of feeding. We have four different types. We have suspension feeders, deposit feeders, fluid feeders, and mass feeders. We fall under the mass feeder category because of the fact that we do have to kill, and then from there we have to do more work, right? So using mastication or chewing, we bite off large chunks of our food, and then we masticate it. We break it down into smaller pieces, and then swallow for our digestive tracts to be able to absorb the necessary nutrients. There's also suspension feeders, which essentially filter nutrients from the air or mostly from water. There's also deposit feeders, which feed on organic material deposited on sediments and on the surface of wherever they happen to live. And then there's fluid feeders, which have to suck up, right, form some sort of a vacuum in order to suck up, for example, nectar from these flowers that are shown there. In terms of locomotion or movement, first the functions. Yeah, so why is it important for animals to be able to move? And some animals need to move large distances in their migratory patterns. First and foremost is to find food. Without food, there's no energy. Also to find mates, to possibly escape from predators, disperse to new habitats in case resources run low within an ecosystem. Ways to achieve locomotion is burrowing, slithering, swimming, flying, crawling, walking, running, and it's all powered by muscle and skeleton. Yeah, so these two working together, some sort of a skeleton and some variation of muscles, their combination and their working together allows for animals to move. And these are, again, things that you should already have knowledge of. Now the small details we'll go over later on, but for now there's at least 3 types of skeletal systems. There's hydrostatic skeletons, endoskeletons, exoskeletons. Endoskeletons is basically what we have, mammals, and a lot of animals contain an endoskeleton. These are hard structures found inside of the body which allow the animal to hold a structure, hold a shape, and also to move. We use bones. Exoskeletons, such as the ones found in arthropods, are essentially made up of some sort of really hard carbohydrate, and these exoskeletons are found, as the name implies, on the outside of the organism. And then hydrostatic skeletons, for example, in worms, some types of worms, hydrostatic skeletons allow them to create some sort of internal pressure within their coelom, or using their coelom. This pressure is formed by the water that is found within the coelom of the species or the animal, and furthermore, they actually have the ability to modify the amount of pressure that they put outwardly into the walls of their body, so that they can modify the movement that they have to make. So in order to move, we need limbs. So animals need limbs. There's different kinds of limbs. We're looking at 4 different types here. We are looking at lobe-like limbs, 2 feet, which are 2 types of limbs that rely on hydrostatic pressure. So then we have jointed limbs. There's also parapodia, and then arms and tentacles, which rely on bones and mostly muscles that allow for the movement of the animal. Keep in mind, muscles rely on a set of proteins called the actins and the myosins, so working together in a sliding filament type of way that allows for movement of the animal. And last but not least for this lecture, we have the last innovation that we see in animals, which is reproduction, the types of reproduction. 2 major types. There's asexual reproduction, as we've talked about in the past. These animals are essentially clones of their parents. So the parent cell will, either divide by binary fission, by splitting, or by budding. But the thing is that the daughter cells, well, they're going to be exactly the same as the parents, so genetically the same. So they're going to have to rely on other means and on other modes to be able to achieve genetic variation, if they do achieve genetic variation. Now, the other type of reproduction is sexual reproduction. We have external fertilization and internal fertilization within sexual reproduction. Fertilization is key to sexual reproduction. Recall that for fertilization to take place, sperm is needed and an egg is needed. There's 2 different ways in which this can be done. First and foremost is external fertilization. So eggs are laid on the outside of the body of the female, and then the male goes ahead and releases the sperm right on top of the eggs in order to fertilize those eggs. So those eggs go ahead and develop into the animal. They're outside of the body of the female. Now, internal fertilization, we can see it in many animals, including including mammals. So there is the need to insert sperm in some way or another into the sperm of males into the egg of the female in mammals. So we've talked about this in the past. We'll look at more details later on next week, or within the coming weeks rather. So the uterus becomes essentially the place where sperm is received, and the place where sperm meets the egg for fertilization to occur. Now, once fertilized, eggs undergo different modes of development or embryonic development. We have viviparous eggs, or species. We have oviparous species and ovoviviparous species. So viviparous species giving birth to live young, as you see in mammals. Oviparous species, ovi refers to an egg, and these are species that deposit fertilized eggs essentially, and these are outside of the body, and the egg is basically nourished by the yolk, so by the yellow part of the egg, and that's how over time this animal develops, again within the egg. Ovoviviparous is still within an egg, and the offspring is still nourished by the yolk, but then the egg is not deposited outside. The egg is kept within the body of the female until it's ready to be given birth to. And last but not least is the different types of life cycles within animals. So there's two phases within the life cycle of an animal. There's a diploid phase and there's a haploid phase. Recall the difference between the two. Diploid phases are phases essentially composed of cells that contain two sets of chromosomes, and haploid cells contain only one set of chromosomes. So in humans, it would be the sperm and the egg, which is the haploid stage of the animal in humans. So the sperm and the egg are haploid because they only contain 23 chromosomes, again in humans. But then after fertilization, that's when the diploid stage takes place, and then the entire duration of the life of the animal is a diploid stage of the animal. Yeah, so I'm showing here different stages within the life cycle of an animal. Evidently, we don't necessarily metamorphosize. Some animals do. So first and foremost is you need fertilization to form a zygote, which undergoes embryogenesis to give rise to a larvae, and then that larvae metamorphosizes into a sometimes movable form called the juvenile, which grows into an adult, and then gametogenesis takes place, which is the formation of sperm, or spermatogenesis, or the formation of an egg. During oogenesis, those two are combined in some way through sexual reproduction to give rise again to a diploid organism. Yeah, so there it is.